As Ann mentioned earlier, this Carl Meyer Award presentation by Dr. Prodi will be videotaped because the NIGMS, as part of their 50th year anniversary, are sponsoring this videotaping. The namesake of the Carl Meyer Award was an eminent glycobiologist in the middle of the last century. Carl Meyer is considered by many individuals as being the father of glycosaminoglycan research because of his contributions to our understanding of hyaluronan, chondroitin sulfate, keratan sulfate. And in 1991, a year after his death, the Society for Glycobiology decided to honor his work and his memory by instituting an award that bears his name. And so the Carl Meyer Award recognizes a well-established, currently active scientist who has made widely recognized major contributions to the field of glycobiology. And in the intervening years, a number of prominent glycobiologists have been recipients of this award. This is a virtual who's who of glycobiologists, eminent glycobiologists. And not only are they great scientists, but I consider a lot of these people friends. They're wonderful individuals besides being just great scientists. Now, last year, the winner of the award was Steve Rosen. Now, regarding this year's recipient, many years ago, when I was a graduate student, I was manning a poster at a ASB&B meeting. And when I was given this poster, some guy walked up to my poster and started asking me a lot of questions about my work. And he was very nice about it, even though I was a lowly graduate student. He was very nice and asking me a lot of questions. And as he walked away, he was very complimentary. And then later on that evening, I was asking my good buddy, Tom Altman, about that guy. And Tom says, well, who is that guy? I said, well, I don't know anything about him, but all I know is that from his name badge, he had this, in his tag, he was from some distant country that was south of Texas. And so Tom says, well, what was his name? And I said, well, I don't remember his name, but I think his name was Louis Laylor or something like that. And Tom says, then Tom, as is his wont, then proceeded to lay into me with some choice expletives and told me that I should have known that that person was the great Louis Loire, who a few years earlier had won the Nobel Prize for the discovery of UDP glucose when he was studying glycogen synthesis. Now, then a few years later, when I became a postdoc, in my opinion, those were exciting times because that was the infancy, the dawn of the topic of N-glycosylation. At that time, there was a lot of prominent laboratories that were working on the pathway and the mechanism of N-glycosylation. In fact, laboratories that were being headed by many of the people that were recipients of the Carl Meyer Award, like Stu Kornfeld, Pamela Stanley, Phil Robbins, Bill Lenars, Harry Schachter, prominent laboratories. But then I started to notice as a postdoc that there were a lot of publications that were emanating from a person in this person's laboratory. And this person, I hadn't met the person at all, but the publications that were coming from, that were either first author or senior authored by that individual were major findings. As an example, he authored the first description of a polyprenol being involved in N-glycosylation in eukaryotes. Polyprenols were previously known to be involved in glycosylation in bacteria. That was the work of Phil Robbins and independently by Jack Strominger. But this individual showed that it was true also in eukaryotes. And not only that, he showed in other publications during that time that UDP glucose was a donor of glucose for the formation of dolichophosphoryl-glucose, which in turn is a donor of glucose for dolicho-linked oligosaccharides, which in turn is a donor of oligosaccharide to asparagine of proteins. These were done with rat liver microsomes. And then not only that, he showed in other publications that this was not true just for UDP glucose. This was true for GDP mannose, UDP NS to glucosamine, or donors of sugars to dolichol as well in N-glycosylation. So, so when he was having all these publications, what was amazing to me was he was doing this work not using sophisticated instrumentation like mass spec or something. He was doing this by simple techniques, using uniformly labeled glucose and then analyzing the products by something called paper chromatography or thin layer chromatography. Just elementary techniques. He was making major observations. And then a few years later, and I kept thinking to myself, who is this guy? And then a few years later, when I had my own laboratory, additional experiments came from this individual, either as a first author or as a senior author. He was publishing that this N-glycosylation using dolichol was not just applicable to rat liver microsomes, but also applicable to yeast in the formation of mannan. But around that time, the consensus was formulating that this dolichol-linked oligosaccharide was universal in eukaryotes as a donor. This glucose 3 mannose 9 2 glucosamines linked to dolichol was a universal donor. But then there was a publication from this guy's lab who said, well, this was not universal. He published a number of articles from protozoan parasites like trypanosomes, Cryphidia, Leishmania, which he showed that non-glucosylated polymannose dolichol oligosaccharides were donors to the asparagine. And not only that, after donation to the asparagine, then glucose was added transiently. And I thought to myself, who is this guy? And then, as a matter of fact, I couldn't believe it at the time. So, matter of fact, in my institution, I went down the hall because there was somebody working on these critters called Leishmania. I never heard about them before. So I got some of these Leishmania organisms to try to see if this guy was correct or not. And sure enough, he was correct. That non-glucosylated polymannose oligosaccharides were donated to asparagine. And then I kept thinking, who is this guy? Why was he so good? As a matter of fact, I thought this guy must be so good that he and his colleagues must be doing experiments which they weren't wearing lab coats, but wearing sport coats. And so, and he was the work coming out of his lab was amazing. And not only that, in subsequent years, he showed, he analyzed the glucosyl transferase involved in the transient glucosylation mechanisms, which we now know is the pioneering work for quality control of protein folding. He also worked in recent years in the oligosaccharide transferase. So as you might imagine, over the decades that he's been working in this field of N-glycosylation, you might imagine he's been the recipient of many awards and honors. And just to list a few of these, he is a fellow of the American Academy of Microbiology. As you might imagine, from his own country, he is a member of the National Academy of Sciences in Argentina, that far distant country south of Texas. He is also in the neighboring country of Brazil, and in a faraway country from him is the United States. He's a foreign fellow of the National Academy of Sciences in the United States. An amazing feat. He is also a member of the Third World Academy of Sciences, and he's also a recipient of the Howard Hughes Medical Institute International Research Scholar. This is just a partial list. And now we can add to this list the Society's most prestigious award, and that is the Karl Meyer Award. So at this time here, I'd like to have all of us welcome Dr. Armando Parodi as a recipient of the 2011 Karl Meyer Award. And in his honor, we have a plaque, and we have a check, which for his. There we go. Thank you, Ahmed.
Well, I want to thank the Society for this award. In my lifetime, as Sam has just told you, I have received a few awards. But this one really, I would say, touches my heart because it represents the recognition of my work on glycobiology by my peers. So thank you very much. The title I chose for my talk, it's a bit queer, and I want to show you that although we are scientists and we think that we control everything in our experiments, I mean the temperature, the time, the presence of ions, and even when we work with live animals, the color of the room where they are kept, etc. I will show that at least south of the equator, there are a few forces unknown to us that we cannot control and that heavily influence the result of our research. My story is, I have an additional title, more serious one for my talk is this one, Protein and Glycosylation as Seen from Buenos Aires, and I must say that I hate boring people with my recollections. The first chapter of my talk, and there are 3 of them, is chance. My story starts in the late 60s, as just some told you. At that time, I was doing my PhD thesis under the supervision of Louis Leloir on the synthesis of heavyweight glycogen. And in those years, the several papers coming from 2 labs, that of Jack Strominger at Harvard and that of Phil Robbins at MIT, showed that polyprenol-linked glycans were intermediate in the synthesis of several bacterial cell wall components. Then Leloir, together with young postdoctor Nicolas Behrens, who had just finished his thesis under the supervision of Enrico Khabib, decided to study if lipid-linked glycans were also these intermediates in sugar transfer in mammalian cells. And what they did was to ask me for some rat liver microsomes I had just prepared for my PhD thesis. They incubated them with UDP-C14 glucose, and they submitted the reaction mixture to chloroform methanol water 3-to-1 partition. And this partition gives you 3 phases: the upper or aqueous phase, where all substances soluble in water are present, then a protein interface, and a lower organic solvent phase, where most lipids are present. Upon doing this to the reaction mixture, they found that part of the label that was initially present in UDP-C14 glucose was present in the lower phase. Not only that, but the new compound that had been formed was labeled under mild-lasting conditions, and this suggested that it could be an intermediate. They then purify by column and thin layer chromatography an acceptor from rat liver that stimulated this reaction, but they didn't get enough material to do a mass spec analysis. But it came to their attention that a group in Liverpool in UK, the group of Morton, had described the existence of polyprenol in mammalian cells. They call it dolicol. Dolicol in Greek means big. This was because this compound had double the size of the bacterial undecaprenol. And so Behrens and Leloir, they isolated this compound according to Morton, and they phosphorylated it using a protocol that had been described for phosphorylation of steroids, and found that this phosphodolicol had the same chromatographic property as the compound they had isolated from rat liver. And not only that, this phosphorylated dolicol also stimulated this reaction, and the product form, dolicol monophosphate glucose, had the same properties of the compound they had initially described. So then they found that upon incubation of dolichol C14 glucose with more rat liver microsomes, a part of the label went from the lower phase to the protein interface upon a Crawford methanol water 3-to-1 partition. So they published a paper in PNAS in 1970. This was the same year when Leloir got the Nobel Prize. The title of this paper was Dolichol monophosphate glucose, an intermediate in glucose transfer in liver. And in this paper, they assumed that this compound that was present, that was formed upon incubation of dolichol monophosphate glucose with rat liver microsomes, was a glucoprotein because it went to the interface and upon a Crawford methanol water 3-to-1 partition, and also because it was insoluble in 10% TCA. I then had finished my PhD thesis and joined the group, and I felt that this identification of these glucoproteins was not very orthodox. What I did was to digest this labeled protein interface totally with a specific protease, and found that upon a second Crawford methanol water 3-to-1 partition with additional carrier protein, the label still went to the protein interface and was insoluble in 10% TCA. When I showed this result to Leloir, he said the magic words, well, perhaps it's not a protein. And it took almost a year to find out what this substance that was formed from dolichol monophosphate glucose 1. It really gave us a very hard time to find what it was. Leloir baptized with the name of cagolin. For respect to the audience, I will not translate this word. Those who understand Spanish know why. So in our papers appears as GEA, or glucosylated endogenous acceptor. One day, we knew that this cagolin or GEA could be solubilized with detergents, but that was not much help to identify what it was. So one day, Leloir was trying different Crawford methanol water mixture in different proportions to see if he could solubilize it with these mixtures. I may say that Leloir was the ideal eternal postdoc, because even being a Nobel Prize awardee, he worked very hard at the bench, at least 8 hours a day until he died. So it was really fantastic. And he found that Crawford methanol water in the proportion of 1:0.3, that's only one phase, could solubilize this product, this compound. And then things went on smoothly, and we described that this compound was a dolichol pyrophosphate linked to an oligosaccharide. And we published this in 1971 in a PNAS paper. In accompanying papers, we described the synthesis of dolichol monophosphate mannose and upon incubation of rat liver microsomes and GDP mannose, and of dolichol pyrophosphate N-acetylglucosamine using of dolichol pyrophosphate N-acetylglucosamine using UDP N-acetylglucosamine as sugar donor. We also published some papers showing that these oligosaccharides had at least 2 N-acetylhexosamine and several mannose and at least 2 glucose residues. The final structure of this oligosaccharide was described by Stuart Kornfeld lab some years later. Then you see, well, evidently this cagolin, dolichol pyrophosphate glycan, couldn't be a final product. So it looked as an intermediate. So what we did was to reincubate it with more rat liver microsomes. And well, the only product we initially obtained was glucose. And the longer we incubated this compound with the microsomes, the more glucose we obtain. So perhaps we thought that the trick to solve this problem was to use very short incubation times. And this is the page of my lab notebook. This is from December 71. I think it's the first assay of the oligosaccharide transferase. Here you see we put cagolin, that is the dolichol pyrophosphate oligosaccharide, labeled in the glucose. In the glucose, we incubated with buffer, manganese, water, enzyme as a detergent, and we submitted this mixture to a chlorformethanol 3-to-1 partition, and this is the counts in the upper phase. That's mainly glucose. This is the counts that it could be extracted with chlorformethanol water 1103 from the protein interface. That is the substrate that went down. And then we have the label that remained in the protein interface. I could not be extracted by this mixture, and this we showed it was the real glycoprotein. This was published in a PNAS paper in '72. In the next 3 or 4 years, several papers came from different labs, mainly even from Leloir's lab, from Bill Lenard's lab, from Ed Heath's labs, showing that incubation of microsomes with GDP manus led to the synthesis of dolichol pyrophosphate oligosaccharides containing 2-acetylglucosamine and a variable number of manus units, no glucose, say 6, 7, 8, 9 manuses. And upon incubation of these dolichol pyrophosphate glycans with microsomes, the glycan was also transferred to protein. At that time, it was assumed that the glycans, the dolichol pyrophosphate derivatives containing only N-acetylglucosamine and manus units, were the real precursors in protein glycosylation because, as you well know, there are no glucoses in glycoproteins. And it was thought that the glucose-containing compounds initially described by Leloir's group was an oddity of no real biological significance. Then in '77, and at the end of '77 and the beginning of '78, 2 seminal papers appear in the literature. Well, these are the reactions we had initially described. Two seminal papers appeared in the literature, one by the group of Phil Robbins, and here is Sam. And essentially what they show in this paper was that the glucose-containing dolichol pyrophosphate oligosaccharide, initially described by Leloir, was the main the dolichol pyrophosphate derivative occurring in mammalian cells, and that it was transferred to protein about 20-fold faster than compounds lacking glucose units. And the second paper is one coming from Stuart Confer's lab, where he showed that the complex type glycans came by processing of the glucose-containing oligosaccharides initially transferred to protein. So these 2 papers really vindicated the earlier work by Leloir's group. And I want to mention you what's one of Murphy's law, that is the ultimate principle that looks like Yogi Bear sentences, because it says that when you are investigating the unknown, you do not know what you will find. Therefore, initially, Burns and Lelay could have used any sugar nucleotide could have been chosen for the initial experiments. Nevertheless, it was the use of UDP glucose C14 glucose that allow the last group to establish the pathway of protein and glycosylation. And the question is, why was UDP glucose, this nucleotide sugar, chosen? And the simple answer is that at that time, the whole institute in Buenos Aires was devoted to study glycogen synthesis and its regulation in mammalian cells. So UDP-C14 glucose was the only available compounds. That's the answer to the question. And, well, then the second chapter of my talk is cumbersome bureaucracy. I then established my own independent group, and the first work I did was to show that this reactions, the formation of dolichol monophosphate glucose, is transferred to a dolichol pyrophosphate oligosaccharide, and is transferred to protein, also occurred in yeast cells, in Cerevisia. Then in 1980, a paper appeared in the literature from a British colleague claiming that this dolichol, this pathway of protein glycosylation involving dolichol derivatives didn't occur in trypanosomatid cells. And the reason for that assertion was that he couldn't synthesize dolichol monophosphate glucose when he incubated microsomes from these bugs and UDP-C14 glucose. And we decided to challenge this concept. And what we did was to incubate live cells with C14 glucose and to isolate the dolichol monophosphate glycans that were formed. And we found that our British colleagues were correct. These bugs cannot synthesize dolichol monophosphate glucose. They synthesize dolichol pyrophosphate derivative containing 2-acetylglucosamine and a variable number of mannose units depending on the species, 6, 7, or 9. And then we decided to, at the time this pathway of glycoprotein processing in the ER has been already established in mammalian cells. Well, then we decided to study how these glycans that didn't contain glucose units were processed when transferred to proteins. And we use Trypanosoma cruzi cells. T. cruzi is the causative agent of Chagas disease, which is prevalent in Latin America. So we incubated live cells with C14 glucose, and we isolated the endo-H-sensitive protein-linked oligosaccharides after only 5-minute pulse with C14 glucose. And when you, as this T. cruzi transfers a MAN9, we expected to find first MAN9, then as it was processed, MAN8, MAN7, and so on. But instead of that, we obtained 3 compounds after only 5 minutes of labeling that didn't migrate on PEG chromatography as the standards of -9, 8, and 7. These 3 compounds, 1, 3, and 5 with Roman numerals, gradually disappeared upon chasing the cell with unlabeled glucose, and the compounds that we thought initially that would appear, that is -9, 8, and 7, gradually appeared. Just by using the traditional old methods of acetolysis, total acid hydrolysis, and so on, we could establish that these compounds were glucose 1 -9, glucose 1 -8, and glucose 1 -7. And so we proposed the following reaction of ER glycan processing occurring in Trypanosoma cruzi, that is N-acetylglucosamine 2, MAN9 is transferred to protein, and this is transiently glucosylated and deglucosylated. That's why the initial compounds we observed were these ones that gradually disappeared upon chasing the cells, and finally we obtained these 3 compounds. The question was that whether this reaction of transient glucosylation of glycoproteins also occurred in mammalian cells. And here things, well, we published these results in a JVC paper in 83. And the question, we wanted to see if the same transient glucosylation of glycans occurred in mammalian cells. Here things are a bit more complicated because unlike what happens in trypanosomatids here, glucosylated oligosaccharides are transferred to protein. So actually there could be theoretically two pathways to form glucose 1-8 and glucose 1-7. One is pathway B is similar to what happens in T. cruzi cells, that is, complete deglucosylation and then transient glucosylation directly from UDP glucose. And the other pathway is when you reach glucose 1-9, then a demanosylation to glucose 1-8 and further demanosylation of glucose 1-7. In fact, the formation of glucose 1-8 and glucose 1-7 had been observed by two different labs. One of the Stuart Confell, when they incubated mammalian cells with C14 galactose, and by Bob Spears at Bob Spears' lab when they incubated thyroid lysis with C14 glucose. But in both cases, as the presence of ER minosidase in the ER was known, they ascribed the formation of these two compounds to pathway A. As we knew that pathway B happened in T. cruzi, we designed the correct experiments and could prove that in vivo, using thyroid lysis or rat liver lysis, or in vitro, using microsomes from thyroid lysis or from rat liver, what happened in mammalian cells was pathway B. In the case of cell-free assays, we found that incubation of microsome with C14 glucose led to the formation of glucosylation of endogenous microsomal glycoproteins directly from UDP glucose without the intervention of Dolichol derivatives. So we proposed the following pathway occurring in the ER of mammalian cells. I mean, this glucose 3-9 is transferred to protein, deglucosylated, and then and demanosylated and transiently glucosylated and deglucosylated. And we published these results in two papers. The first is one JVC in 83 and this in 84. This is transient glucosylation of protein-bound -9, 8, and 7 in calf thyroid cells, a possible recognition sign in the process of glycoprotein. And this paper we described the same reactions occur in rat liver and plant cells. Now, going back to the, going back to the first observation that led to this research, that is the incubation of live Trypanosoma cruzi cells with C14 glucose. We repeated this incubation using not C14 glucose, but 2-tritiated mannose, because it seemed to be a more logical precursor to incubate cells, because the glycan transfer that doesn't have glucose units. But using 2-tritiated mannose, we obtained a different result. The first compound that appeared was that was initially thought we would see -9, followed was by -8, -7, and -6. Why this difference? Well, the reason for the difference is that the amount of monoglucosylated compounds is minimal compared to that of unglucosylated glycans. And as you well know, when 2-tritiated mannose is transformed to glucose, it loses its label. Moreover, at the very beginning, conversion of C14 glucose to mannose takes some time. So in very early times, the 100% of the label here, of the 5 minutes of labeling, is in the glucose units. So the use of C14 glucose and not of 2-tritityl mannose allow description of the transient glucosylation process. And why is that we use C14 glucose and not 2-tritityl mannose? It was because 2-tritityl mannose has a higher specific activity than glucose and is cheaper. But if at the late 70s and early 80s, it was really a nightmare to get label stuff through the customs in Argentina. It was incredible paperwork we had to do, and we had to get the approval of, I know, many different officials. So what we did was to buy label stuff that we thought we would need in the next 4, 5, 6 years, to keep it in the freezers and to use it according to ongoing research. Now, the half-life of tritium is only 13.5 years compared to 5,000 years for C14 glucose. And also, 2-tritityl mannose has a high specific activity, so it's decomposed by radiolysis. So we opted for the C14 compounds that it has a lower specific activity and is much more expensive because of this restriction of the local custom. So the invaluable but unsolicited help from the local custom is greatly acknowledged. And my idea is that cumbersome bureaucratic regulations are active contributors to science development as they promote creation of new neuron circuits in investigators eager to evade them. So the last chapter of my talk is wrong ideas. At the end of '84, there was a meeting of the Pan-American Society for Biochemistry and Molecular Biology in Buenos Aires, and I was in charge of organizing a symposium on glycobiology. And for the meeting, I invited Harry Schechter and Stuart Kornfeld. I remember that taking Stuart from the airport to his hotel, he kindly suggested me that to fully convince the glycobiology community of the occurrence of this transient glucosylation process, I had to purify this glucosyl transferase. Of course, for that, I couldn't rely on endogenous microsomal glycoproteins. I had to have an exogenous acceptor glycoprotein, and he suggested me to try thyroglobulin. As soon as the meeting was over, I bought thyroglobulin from Sigma. I incubated it with rat liver microsomes, UDP glucose, and nothing happened. I tried different conditions, temperatures, ions, detergents, even no detergents, but trying to disrupt the ER vesicles with sonication, and nothing happened. So I put this project aside. From time to time, I retook it with negative results. 4 years later, in '88, I remember an old paper from Bill Lenard's lab that was called Enzymatic Conversion of Proteins to Glycoproteins. In this paper from PNAS from '77, what Bill did was to, alpha-lactalbumin has a hidden sequence, glycosylation sequence, asparagine X threonine, I think, that it's not used in vivo. However, upon denaturation, the protein becomes an acceptor of glycans from tolycol pyrophosphate derivatives in cell-free assays. So I thought, well, perhaps what happens in thyroglobulin is that the whole glycan, -9, the acceptor, is hidden inside the protein moiety. I immediately dismissed this idea because glycans are very hydrophilic compounds that have to interact with water. Nevertheless, in desperation, I tried this idea, and it worked. It worked. And this is my lab notebook. That's in June '86. This is the incubation of rat liver microsome with UDP-C14 glucose, and this is this amount here represents the glucosylation of endogenous microsomal proteins. This is the addition of native thyroglobulin, and actually it inhibits the reaction. But this is the, what I did is to take a thyroglobulin, the nature is with 8 molar ureas, take out the urea and use it as an acceptor, and it really worked. And the first surprise by the result was me, because I thought that everybody said that the glycans had to be facing the water dissolved even under native conditions. So I was really surprised, and I did an additional experiment that I compared the, so if this were true, I mean, my initial idea was true, the best acceptor would be just a free glycopeptide, that is a -9 linked to a single amino acid asparagine, because this would be really readily accessible to the glucosyl transferase. But that was not the case. The free glycopeptide was not an acceptor. This is the comparison of the acceptor power of the same amount of glycan linked to asparagine or linked to the nature thyroglobulin. So you see that thyroglobulin the nature thyroglobulin is a much better acceptor. So this showed that upon the denaturation of thyroglobulin, what I was doing, it was not the exposure of the glycan, but modifying the conformation of the protein moiety, and that's what allowed the reaction to go on. I published these papers, these results in publication in '89 and '92. This two in biochemistry, this is JVC. This is the final purification of the glucosyl transferase in '92, 8 years after Stuart had suggested me to purify the enzyme. So the conclusion of this third chapter is that well-documented results in the literature should be followed, but not too much. Room for wrong ideas should be left also. And I think I have shown you that in 3 cases, the use of UTP glucose, the use of C4, and not other sugar nucleotides, the use of C14 glucose and not 2 treated mannose, and the adoption of the correct wrong idea and the test of the correct wrong idea and against all logical were not rational decisions, were imposed for someone else, and nevertheless these decisions were highly helpful for my research. And my idea is that the bit of irrationality that still permeates in our endeavors is that what makes them really interesting and exciting. And to end with, I want to mention 3 colleagues whose work was really inspirational to my own research. And this is, the guy here is Leloir. He was a fantastic guy, very smart, a gentleman and a very good friend. And this handsome young man here is me. This photo was taken 45 years ago. These 2 guys, you know them very well. This Phil Robbins and Stuart Confell. Their work and the work of their collaborators in the late 70s and early 80s was really fantastic. So I thank you all very much. And this guy here is Harry Alenius, that in the early 90s, he put my pure glycobiology work within the right context of cellular biology when he showed that this glucosyl transferase was a key player in the quality control of glycoprotein folding. So my last slide is this one. Thank you for your attention and the award. and also for the very generous American funding from NIH and Howard Hughes Medical Institute. Thank you very much.